How did people possibly know these things? I mean, it's easy. You've seen this. It's in the book. But how could you have known these things? How did anybody know? How did Crick know that the genetic code was not this loopy, funny thing that was binding the amino acids, but were these triplets? By the way, he didn't have DNA sequencers available. Nobody could sequence DNA. How could Crick have known it was something else? I'll tell you, Crick and 3 other people wrote one of the most beautiful papers in molecular biology. Crick, Brenner, Barnett, and Watts-Tobin wrote an amazing paper called On the Triplet Nature of the Genetic Code, which they wrote before they knew the genetic code. And before certainly before anybody could sequence DNA, they knew it was a triplet code. How could they possibly know this? Well, I'm going to tell you very briefly, and if it doesn't make sense, forgive me. They used mutants. Basically, they found mutants of a particular bacterial virus that had the effect, they didn't know for sure, but they kind of suspected this, of causing one letter of the DNA not to be changed, but to be deleted. Let's think about what happens if a letter of the DNA is deleted. What's going to happen to our translation here? It's going to screw up everything. Suppose I deleted this C here. Sorry, let's delete this G here, okay? Now it's going to read AAC, CCC, etc. Every single, we call these codons, every codon will be screwed up after that point. So these are codons. So if I delete one letter, it screws up every codon past that point. We call that a frame shift, because this is the reading frame. Here's our reading frame. And what it does is it throws us out of our reading frame. And so it's called a frame shift. What do you think that'll do to the protein if every single amino acid past that point is something else than it's supposed to be? It's going to destroy the protein. They collected a bunch of these mutants that they had reason to think were deletions, and then they crossed them together. And these were phage, and you could do genetic recombination crosses. And they made double mutants that ended up putting the 2 mutations on the same phage very close to each other. Remember, they could do genetic mapping. They knew where things were, even though they didn't know about any of this. And when you made the double mutant, what do you think it does to the protein? It shifts over 2, and what does that do to my code? It still screws it up. But if I were to cross, tell me, if I cross 3 mutations onto the same phage and they're right nearby, a deletion, maybe pretty close by another deletion, and pretty close by another deletion, when I get past those 3 deletions, I'm back in the reading frame. And they found that 3 harmful mutations, 3 totally harmful mutations, when combined, basically worked fine. 2 didn't, 1 didn't, 3 did, it's multiples of 3. I gotta say, it gives me goosebumps, that paper. It's one of the most beautiful papers. They knew it was the triplet code just from pure genetics. It's a gorgeous paper. Of course, the geneticists like me love that paper. The biochemists, they say, that's great, but what's the code? Please tell me the code. It's great you guys are just having a wonderful time that it's triplets, but tell me, what are these triplets? So then that took biochemists. Biochemists come along, and they somehow have to actually work out what the code is. They have to do an experiment with like chemicals and grinding things up. So they get in there and they grind something up. They grind up cells, and they want to translate RNAs. So Marshall Nierenberg takes an RNA that he makes. Turns out there was this trick he had for making an RNA consisting of all exactly the same nucleotide. There's a special enzyme you don't have to know about that would stick together nucleotides, and if you just gave it, say, U, you would get an RNA that said U. And when he threw it in with an extract of the cells, he could get it to translate U into phi. He got polyphenylalanine from polyuracil. And what does that tell us about the codon for phenylalanine? It's UUU. Let's go back to our genetic code here. UUU phenylalanine. What else can you get me? How about CCC? CCC. Polyproline. How about AAA? AAA. Lysine. Nuremberg got it. Bingo. He had those. But it's actually only the homopolymers. What about if I wanted ACA? What do I do? See, Nuremberg didn't have a way to make ACA. He could throw at his enzyme a lot of U's and he would get U's. But if he threw A's and C's at his enzyme that would stick together nucleotides, what would he get? No, wouldn't get just one or the other. He'd get random things. It could go AA, CA, CA, CCA, CA, CA. He would get a probability distribution of all the possible orders of A's and C's being stuck together. And when he fed that in, he would get the protein that came out having all the possible codons that had A's and C's in them. So he didn't learn nothing. He actually learned, let's see, A, C, A, threonine, C, A, C, histidine, C, A, glutamine. He would begin to collect that A's and C's must encode those amino acids. And then he could try other mixtures of things and other mixtures and tweak the concentrations, and you could begin to collect information. Better, though, would be if you had a real super-duper chemist who could make you A, C, A, C, A, C, A, C, and not be playing around with these random probabilities. And that's where a former colleague of ours who passed away not so long ago, Gobin Khorana, came along. Gobin, who taught here at MIT for many, many, many years, was the super-duper chemist who figured out how to, in a disciplined fashion, get A, C, and also how to make triplets. Between them, Nuremberg and Gobin Khorana cracked the genetic code and gave us the whole thing. And it's pretty cool. In fact, we have a tape on the website of Gobin before he passed away talking about his childhood in India and his schooling, which is maybe different from some of your schooling. It consisted of a box under a tree and people sitting around learning under a tree there in his village in India. But he went on to share the Nobel Prize with Marshall Nuremberg for cracking the genetic code. So, all right, pretty cool stuff. That's how people know what the genetic code is. Now you just look it up in the book, but it was a wonderful puzzle at the time.